Today's the 14th, so we'll be learning about meiosis. Meiosis is pretty short. It's a little bit like if I understand cellular respiration well, then photosynthesis goes fast. If I understand mitosis well, meiosis goes fast. And then we'll start a little bit into Mendelian genetics. The stuff we start today in Mendelian genetics, I promise you'll find it interesting. It's really good. And then next class, which will be Thursday for us, the last class before break, we will take our quiz. Our quiz basically stops here. So it stops at like controlling the cell cycle. So it's got background genetics. That was like that day where we had like genomes and karyotypes and trisomy 21 and trisomy 13 and stuff. Cell cycle, that's like G1, S, G2, mitosis. Mitosis has PMAT, prophase, metaphase, anaphase, telophase, cytokinesis. And then cell cycle control, that was most of last class where we had like, on the board, I probably wrote it right about here, like cycle-independent kinases and cyclins and proto-oncogenes and tumor suppressors. So today we'll start meiosis and Mendel, but again, that's not on your quiz. So everything that you need for the quiz basically ended with last class. All right. If you're going to be gone on Thursday, I've actually already got the quiz printed. You can take it before you leave. Technically, you can wait and take it until after you come back. But you know you don't want to do that. Because then it'll be like Thanksgiving break, and you have this thing hanging over your head, and you're like, I should probably study, but instead, I just won't. I'll procrastinate, and I'll feel guilty. But if you take it before you leave, then, and if not, then you should. But if you take it before you leave, it's like, you're like, it's done. I don't have anything that I really have to do for bio over break. Yeah, we have a test later on, but that's like a couple weeks after we get back. So, any questions about that? Yeah. For the quiz, you don't need 7.4. 7.4 is meiosis. So 7.1, 2, and 3 are all that matter. Probably like mitosis and cell cycle control matter the most. That'd be like 7.2 and 7.3. Yeah, but it also just depends on what you feel like you struggle with more. I
was reading like 7.2, I think. It was talking about prokaryotic cells too. Should we know that?
No. In 7.2, if it talks about prokaryotic cells, all you'd need to know is that they don't do mitosis. So they do this other thing called binary fission. So as long as you know that they like divide differently, you're good. Yeah, very good question. The question was, if you didn't hear it on the mic, was, hey, there was some stuff about prokaryotes. Like, we didn't talk about that in class. Is that fair game? No, just know that they don't do mitosis the same way. Any other questions? Cool. All right. Meiosis then. So for meiosis, first, let's start with something just kind of interesting. It's always good to start with something kind of cool. Since we're kind of starting genetics, cell cycle involves genetics, Mendelian genetics, of course, does, and then our next unit is titled genetics. There's just some like interesting things. Since we started naming genes and stuff like that, that's a fruit fly. Now, you probably haven't ever looked at a fruit fly up close. They're teeny tiny, but if you magnify them, that's a mutant fruit fly. Doesn't he look all spiky? He's not supposed to look spiky. So he has a mutation in this gene. And so they have like 5 model organisms for genetics. So they study fruit flies, mice, this one type of worm, Arabidopsis, a plant, and yeast. So all the other types of organisms, if you're a genetics person and you discover a gene, they have like all these rules in place for how you name genes. But if you're a fruit fly geneticist, the rule is you can name it whatever you want. So they get to be more creative. So if I discover some gene in this worm, based on what it does and everything else, it'll be like, well, that gene's called ERB72. And you're like, that's not very fun. But if you're a fruit fly geneticist, you can name it whatever you want. Some of them are cuss words. Some of them reference South Park. Some of them reference Monty Python movies. And so this mutation is called the Sonic Hedgehog mutation, because when a fruit fly has that mutation, their hair looks spiky, like a hedgehog. And so it's the SHH, sonic hedgehog gene. And it turns out that the hedgehog genes are found in all living things. So they might not do the exact same thing in all living things, but Sonic Hedgehogs, that's like a Sega character. That's Sonic the Hedgehog. Do you guys know what, do you know who's, okay, I didn't know. Like my generation, we grew up with it, but your generation, I'm not if you're just like, shut up, old man. We don't know what you're talking about. Is that Elmo? Or I don't know. So Sonic Hedgehog genes. And so what will happen is the SHH gene, the Sonic Hedgehog gene, is what they call like a master gene. So just to give you, this doesn't relate to meiosis. This is just some genetic stuff that's related to this unit, kind of. So let's say that it's time to like build an arm. Is there ever a time where I would turn on the genes in a normal human, where I'd turn on the genes for grow a pinky, if I didn't also grow like wrist bones? No, so if I grow a pinky, I should also grow this and this and this and this, these bones, an ulna, a radius, a funny bone, a humerus. And if I grow those, shouldn't I grow all the tendons and muscle and fat and skin that goes on it and all that? So the Sonic Hedgehog genes are master genes. Whenever that switches on, that turns on a bunch of other genes, which turn on a bunch of other ones. It's almost like they're the CEO of a company. If the CEO of a giant company says, we now do business this way, then all of a sudden, like, all employees change what they're doing. Like, we're not doing this on paper anymore. This process is now digital. All right, so now like thousands of employees like change what they do according to the boss. Well, turns out the Sonic Hedgehog genes are in this family called Hox. You don't have to know the name, and they're found in all living things in the same order. So on a fruit fly, you have this gene, this gene, this gene, this gene, this gene, this gene, and it determines the overall shape of a fruit fly. Those same genes in the same order determine the overall shape of a mouse, determine the shape of a human, determine the shape of a dog, determine flowers. So what will happen is I'll have a master gene that's like arm goes here. And if arm goes here, I'll turn on thousands and thousands of genes that grow all these bones, all these muscles, all this skin, all that stuff. If I have a master gene for like, you know, skull or whatever, then like if I flip that on, it's going to activate all the genes to build a complete skull. There's never a time where it'd be like make one cheek bone and that's it. That doesn't make any sense. So it's a master switch. Sometimes you turn on the master switch in the wrong area. So I turn on the master switch for grow an arm right here, and then I turn on the master switch for grow an arm right here. What if I turned on the master switch for grow an arm right here? I would have a complete third arm growing out of my chest. If I turn on the master switch, it's going to grow like everything right there. It's kind of like if I hit go on the moving assembly line for like build a car. When I hit go, it goes like all the way to the end. If I hit go, like there's going to be a complete car. So what they find is crazy stuff. So here's a normal fruit fly. Looks kind of like a bee, but it's a fruit fly. This one right here, they call these aristae. Don't worry about the word. You and I would just call them antenna. But those are aristae. Those are normal aristae. So what scientists did is they're like, all right, we think we found the master gene switch for legs. So we're going to turn it on there and there. So now instead of growing two antennae, it'll grow two legs out of its face. So those are legs. Just like I was saying, if we turn on the master switch for grow an arm right here, then I'll like grow an arm out of the middle of my chest. It won't work right because for my arm to work, I need like a clavicle and a shoulder blade and like the joint in my shoulder has to line up. So it'd just be like this dangly, like I wouldn't have like this cool, it wouldn't be like I'm working on something and I need like a third hand to hold like a board or whatever I'm sawing or whatever. And all of a sudden I'm just like, well, let me unbutton my shirt and like, you know, like that, or that'd be freaking awesome. But they just turned on those switches. So every single cell in my body has a full set of genes. So if I have a skin cell right here, it has the genes for growing brain, lungs, heart, skin, and everything else. But only the skin genes are on. If I take a gene from my brain, if I take a cell from my brain, that brain cell has the genes for brain, heart, lung, skin, but only the brain genes are on. So I can go in anywhere, and if I turn on the master switch for grow a leg, you'll grow a leg because you have all the instructions for growing a leg. There you go. Normal fly on the left, double torso fly on the right. You're only supposed to have like one torso. It's kind of like head, torso, abdomen. They call it a thorax. We would call it a torso in a human. And so they just like doubled it. Thorax, thorax. That'd be like me. I'd have like legs, a waist. I'd have all this with a set of arms, then I'd have like another one. If I was a centipede, I'd just do that over and over and over and over and over and over and over again. I just turn on that master switch again, and now I have a centipede. It's like these flies are alive. Isn't that weird? You like add a torso and wings, and can you imagine just like taking here and here and just like adding another layer on? How bizarre that would be. Be a great fighter. And you got like 4 arms. Eyes are red. Notice anything about his feet? They turn on the eye genes on his feet. So he's got eyes growing there and there. And it looks like maybe even there. Those are his disheveled wings. A lot of times in labs, you don't want your fruit flies flying away, so you just make them all mutant to where their wings are vestigial. They're like crinkled up and they can't fly. It's almost like I'm a penguin or whatever. It's like, yeah, I got wings, but like they aren't good enough to fly. So those are eyeballs right there. Now they don't work. You know, he couldn't like go and like look around the corner or something like that. It'd be freaking awesome. Because now for that eye to work, I have to like have nerve cells that join to the brain, and the brain has to interpret and all that. But those are like eyes. Like if you dissect those, you're like, those are eyes on his feet. Do you guys know that flies taste with their feet, by the way? Their taste buds are on their feet. So when they land on something, whether it's like honey or like a turd or whatever, and they're stomping around, they're tasting it. And they don't have jaws, so if they like it, they throw up on it. Let the throw up kind of digest, just like whatever's in my belly right now. Like if I take food and I put it in my belly, I digest it, right? But what if instead I take all the stuff that digests food and I just barf it out? Then I wait a little while, and then I have a little proboscis like a straw, and I just slurp it up. So if any of you guys are like fruit fly geneticists one day, I need to know the timing of this. If I see a fly like land on my drink, I might get ready to swat him away. But what if he just barfed on it, and then I swat him away, and I'm like, I got rid of the fly, and you're like, you're eating fly barf. So I want to know how long does it take them to like barf and slurp it up. So then I know when to swat them away. It's like the 5-second rule. It'll be like the 3.5 fly second rule or something like that. I don't know. So anyway, these are the things that I want to know in life. Here's that fly where they put the legs on his face. Normal antenna, 2 legs. I'm sure he's completely confused. You know, you just be like that. Sort of like the current hair trend with males. You just like that. And then again, the same genes are found in flowers. So this is a flower. This is supposed to be a flower, but they accidentally turned on the leaf genes. So it's not a flower. It's not made of petals. It's made of leaves. But it's in the shape of this. It's the same genes. I can take the Hox genes that make a flower and splice them into a mouse, and they work because they're the same. So weird. Genetics is so bizarre. So anyway, I couldn't just not show you those things. It doesn't really fit in any particular place. Oh, I got one more. Hang on. So this is a chicken wing. You might say that looks like a human arm. Well, all the bones are the same. I used to teach anatomy way back in the day. For like 5 years, I guess maybe my third through 7th year of teaching, I taught anatomy and physiology. And we spent like 7 weeks dissecting cats at the end of the year. And so I'd have students, they're like, hey, this is interesting. Like we're learning all the muscles of a cat or whatever. Mr. Cotton, why aren't we also like at the same time, like learning the muscles of the human form? Like right now we're learning all the muscles of like this cat form. I'm like, you just did. And they're like, what do you mean? I'm like, they're the same. They're all the same. So what's this in a cat? Brachioradialis. Well, what is it in a human? It's brachioradialis. What's this? This is flexor carpi radialis. What's it in a human? It's flexor carpi radialis. Where is it? Same spot. What does it look like? The same. It's the same. Now, some things are weird because cats walk on all fours, so their proportions, like some of their leg muscles on the hind legs are slightly different, but like they're the same. So if you look at a chicken wing, they've got a humerus and a radius and an ulna and wrist bones, and you know, it looks like a hand like that. So what they did here, bless you, was they turned on some of the sonic hedgehog genes that say like grow a hand, and then they grew a mirror image one on there also. It's like double chicken wing, like that. Buffalo Wild Wings did this. Now you get twice as many wings. That one's a joke. So they really did grow this. But what I would do is I'd make mutant chickens with like double wings, and then I'd open up my own wing stop or whatever, and I'd put everybody else out of business. Because like, how does he get the wings so cheap? It's like mutant chickens. For like chicken wings? It's their forearms, because I can like, when you eat it, I can see the radius and the ulna. So it's their wings. No, if you extend it out, it's like just the bone. Like when you eat a wing, there's like two bones right there, right? They also do like the little legs. Like there's two bones. Those are the two bones of their forearm. It's those two bones. Now, they might not serve you that part, so it might just be like part of the wing, like the forearm part, kind of like my arm. If I was like, I'm serving arms or whatever, I might only serve the bottom half. Maybe they don't have the humerus. I'm not sure. But I know for sure when I eat chicken wings, I can identify, because when I eat it, I'm like, radius ulna. Other people like, this is spicy, and I'm like, look at that radius and that ulna. So, super bizarre. But I thought it was kind of cool. All right, back to our regular schedule, meiosis. Okay, so I've got gamete cells and somatic cells. Some more vocab for us. What were gamete cells? Like, what do we commonly call gamete cells? No like student calls them that. What are examples of gamete cells in humans? There's two. One's from the mama and one's from the papa. Sperm and eggs, not to be confused with ham and eggs. It's different. That's kind of gross. I wish I could take that one back. Seemed funny in my brain, and then it exited my mouth, and I was like, no. So gamete cells, they're also called sex cells. And for example, in humans, that would be like sperm cells if you're a dude, or egg cells if you're a lady. Sperm and eggs. So gamete cells. Somatic cells are everything else. So they are just not gametes. So a brain cell, a lung cell, a liver cell. If whatever I say is not sperm or not egg, then it's a somatic cell. What about skin? Somatic cell. What about liver? Somatic cell. What about sperm? Gamete. What about egg? Gamete. Anything else is going to be a somatic cell. Now, when you get into other types of life, other kingdoms, you get into some other things like spores and crap like that. But most things that we care about reproduce sexually, so they make sperm and they make eggs. Even flowers. Most plants reproduce with sperm and eggs. The flowers are the reproductive parts, and like right in the middle, you have the female reproductive structures with eggs in it, and then pollen is sperm. So the pollen like has sperm in it that swims down and like fertilizes the flower and makes seeds. So gamete cells versus somatic cells. With somatic cells, in order to make new ones, I will use mitosis. And then cytokinesis. If I'm looking at gamete cells, what was the other one? It's not mitosis, there was another osis. Yeah, meiosis. In meiosis, they decided we should have three vowels in a row, M-E-I-O-S-I-S, and then they also do cytokinesis. So somatic cells, they divide by mitosis and cytokinesis. Gametes, they divide by meiosis in cytokinesis. And again, if I understand mitosis really well, meiosis is pretty easy. We basically just have like a couple of slight differences. If you are wondering where this would be in the notes, you just kind of keep on flipping. Eventually you get to a page that literally says AP Biology, Chapter 7.4, Meiosis, like right there. So you go past the cancer stuff, and then eventually, like at the very, very top, it says AP Biology, Chapter 7.4, Meiosis. And there's just a bunch of pictures out to the right. So it looks like that. Okay, cool. So if I'm thinking of mitosis, I'll just kind of do sort of my version of this drawing. I've got a cell up here, and if I have a chromosome 1 from mom and a chromosome 1 from dad, is that diploid or haploid cell? If I have pairs of chromosomes, do you think it's diploid or haploid? Di means two. Yeah, so diploid, diploid. So if I have a chromosome 1 from dad and a chromosome 1 from mom, then that's diploid. And they represent diploid by 2n. So that just means you have pairs of chromosomes. How many chromosomes do you have? 23. All right, 2 times 23. Oh, you're some other animal and you don't have 23 pairs, you have 40 pairs, then okay, 2 times 40. So n is just like how many is normal for that animal. But it's just letting me know I have pairs of them. Just like in math, if I have 2n, that means I have 2 of everything. So I've got 2n, diploid. So I've got a chromosome 1 from dad and a chromosome 1 from mom. All right, cool. Over here, in meiosis, I start off and I'm diploid. I start off the same way. You're going to notice that I put them side by side. I do that on purpose. They also start off diploid. All right, cool. Let me see how this looks. Yeah, if that's big, I think it's fine. I don't think I need to turn on the lights. Yeah, that's fine. Okay, so over here, diploid and diploid. In mitosis, I'll end up going through an S phase. What looks different in my picture after the S phase? If I think back to my cell cycle diagram, what looks different after the S phase is done? Yeah, I double everything. There's 2 of each one. So the way that we would start off in class is we'd be like, I've got a chromosome 1 from dad and a chromosome 1 from mom. And then by the time I get to the end of the S phase, I double them up. So over here, I've got my sister chromatids that I just made. Got my sister chromatids that I just made. All right, cool. So along in here, I went through an S phase. So I started like that, homologous chromosomes. I go through an S phase, and now I have copies. What was it that was holding those sisters together again? What vocab word was that holds blue with blue? Yeah, centromere. Good. So centromeres hold the sister chromatids together. All right, then, eventually, by the time I finish up mitosis, once I'm done with mitosis, I'll have 2 cells, and I'll have split them apart. So I'll have blue and blue. I'll have red and red, just like my original ones. So I made exact copies, hopefully. If I did everything right, then I'll have exact copies. I might have had some mutations, but I'm trying to make exact copies. Does that make sense? I lined everybody up in the middle in metaphase. I pull them apart in anaphase, rebuild everything in telophase, cut the cells in half in cytokinesis, and I've got 2 identical cells. Those look exactly like this. So this cell is still diploid, and this cell is still diploid. And hopefully they're copies. Hopefully they are identical copies. So that's what I do with somatic cells. I do mitosis and cytokinesis, line everybody up in the middle, pull them apart after I copied stuff, and I'll have identical cells. So the goal is identical copies, or identical cells, however you want to say it. That is my goal. I want them to be exact copies. It's a Xerox machine. So in mitosis, followed by cytokinesis, I'm trying to make identical copies. All right. In meiosis, it's different. I want genetic variety. So why is it that you want, if you've got some guy's sperm or some lady's eggs, why do you want every one of his sperm to be slightly different? Why do you want every one of her eggs to be slightly different? Why is that good for the population? What if like dad only made one kind of sperm and mom only made one kind of egg? And pretend that we're like animals that have like litters or something, you know, like we have like dozens of kids and they're all just clones. Yeah. True. True. So hopefully my kids won't mate with each other. Hopefully they'll find someone else's kids so we can avoid the incest thing. Why do I want genetic variety? Huh? Yeah, it's from a natural selection standpoint. If I have 30 kids and they all have some sort of major genetic defect, and that means they're all going to die by age 8, then like all of my kids die. But if they're all slightly different, then somebody is going to be immune to COVID. Somebody's going to be immune to HIV. They found a population in Europe where about 6% of this particular population is immune to HIV. So pretend that we never came up with a cure. Pretend that it was just getting spread everywhere in the world. Somebody would survive it. The flu. Let's say you don't get a flu shot this year, and you never get the flu this year. Is that because you're immune to it, or just because you're really good about washing your hands and stuff? You'll never really know. So if we're all slightly different, then whatever the world has for us in the future, somebody can probably handle it. Does that make sense? If we're fish living around in a stream somewhere, if some of us are bigger, some are smaller. Some are bigger, so we can be more intimidating to other fish. Let's say there's some sort of really good predator out there. Well, now maybe the small ones can go and hide in small areas. So it might be an advantage to be that. Some are brave. They go out and fight. Some are scaredy cats, so they like hide and let everybody else kill each other. Then they come out and they survive. So as long as we have genetic variety, somebody in the human population can probably survive this bacteria or this virus. We're not immune to everything. Nobody's going to survive, you know, like nuclear explosions or like fire or something. It's like, well, if we're all a little bit different, somebody could survive lava. No, nobody can survive lava. But somebody could survive COVID-19 strain 30. That's the idea. In fact, immunologists estimate that any given disease, about 1 or 2% of the population is immune to it, to like that strain. So whatever it is. Ebola, you're like, I've heard Ebola is really deadly. Yeah, it kills about 95% of the people that get it, which means 5% of the people that get it don't die. So it's possible that their immune system is just different enough to where like they're okay. It could be luck, or it could be that their immune system has some sort of genetic mutation that helps them. So with gametes, we don't want identical cells. With gametes, our goal is genetic variety. Genetic variety. All right, cool. So we'll learn in a second. There's 2 ways that we establish that, and those are the 2 main ways that meiosis is different from mitosis. So I got this cell up here. My goal is I want my offspring to all be different. And then do I want my final ones, like my eggs if I'm a girl or my sperm if I'm a boy, do I want them to be haploid or diploid? Sperm, eggs, haploid or diploid? Haploid. How come? Yeah. Yeah, because that way, like, why do I have a chromosome 1 from dad? Because I got a haploid sperm from dad. I got a haploid egg from mom, and that's why together they are diploid. So I'm wanting to make sperm that would look like this. It'd have like a chromosome 1 in it, a chromosome 2, a chromosome 3, and so forth. I'm wanting to make eggs that look like this. I have a chromosome 1, a chromosome 2, a chromosome 3. So one night when mom and dad love each other very much, as I always say, you laugh at me saying that, and then one of these days you might have kids. They'll be like 10, and your wife's going to be like, hey, it's a boy. That means you're up. You got to have the sex talk. You'll be like, how do I say this? And then all of a sudden, in your brain, you'll hear me, and you'll be like, one night when mom and dad love each other very much, a sperm meets an egg. Okay, so this sperm meets this egg, and then that makes the first cell of you. So now you've got a chromosome 1 from mom and a chromosome 1 from dad. That means that that sperm has to be haploid, 1N, and that egg has to be haploid. It has to be 1N. Now, I think the word haploid is kind of dumb. I would have called it uniploid or monoploid, something that implies 1. However, haploid, I can think of it as like half as much as diploid, if you want. But yeah, what it really means is 1 somehow. So haploid cells, they just have 1 set of chromosomes. Diploid cells have 2 sets. So somehow, in meiosis, I want genetic variety and I want haploid. Otherwise, I'm going to have freaky kids, right? If dad's sperm have 2 sets of chromosomes in them and mom's eggs have 2 sets of chromosomes, then that kid's going to have 4 sets of chromosomes. And then he's going to go on to have some kids. So his sperm would have 4 chromosomes, and maybe his wife's have 4 chromosomes. Then their kids would have 8. So every generation, you just get more and more and more chromosomes. Is it okay to have extra chromosomes? Didn't we learn that that was like a problem? If I had like an extra chromosome 13 or a 1 or a whatever, that was bad. So I can't have extra ones. That's what cancer cells look like. We saw that. So I have to somehow get genetic variety, and I want them to be haploid. So I start off, and everything's the same. I'll go through an S phase. Now these guys will have sister chromatids, like that. The only difference is I'm just lining them up side by side. All right, now, they still have the same names for phases. So in meiosis, you still have prophase, metaphase, anaphase, telophase. So it turns out, during their prophase, these chromosomes line up next to each other and they join, and they start trading DNA. We call that crossing over. So the blue ones and the red ones will actually line up side by side. They're not head over heels like these. They're side by side. And so crossing over, if you're looking for like a quick, easy definition, is trade DNA. Crossing over. I'll give you a second. Looks like you're still writing. So in mitosis, I've got a chromosome 1 from dad, a chromosome 1 from mom. I go through an S phase, and I make copies. They line up what I call head over heels. And then I'm going to separate them apart in mitosis and cytokinesis. These 2 cells will be identical. Each one will have a pair of chromosomes, so it's diploid. Over here, everything else starts out the same. Chromosome 1 from dad, chromosome 1 from mom. When I go through an S phase, I copy them. Except instead of lining up head over heels, they line up side by side. When they line up side by side, they start breaking off parts of their chromosomes and trading. And this usually works out okay. So like part of this red one will break off, part of this blue one will break off in the exact same spot, and they'll trade. So they're trading DNA. They call that crossing over. The name makes sense. So what it'll really look like is this. This guy won't be pure blue anymore. He'll have a little bit of that red chromosome on him. And this guy right here won't be pure red anymore. He'll have a little bit of that blue on him. They literally just broke off corresponding chunks. It'd be almost like you and I line up side by side, and I'm like, let's trade arms. And we like take off our arms at the exact same spot. I give you my arm, you give me one of your arms, and now I'm like not a pure Mr. Cotton. So I do crossing over. Then I go through all the other phases, metaphase, anaphase, and telophase, and all that, cytokinesis, and I split. So I'll have two cells here that look like this. Over here, I got this guy, a pure red, and then a little bit of blue in there, like that. Okay, so I did crossing over where they trade some DNA. Eventually, they'll go through like metaphase, anaphase, telophase, cytokinesis. But right now, am I ready to make this into a sperm? No, if it's haploid, doesn't it need to look like that? So don't I still have like twice too much DNA? So what do I need to do? Yeah, I just split again. So if I have twice too much DNA, I just split it in half. So I divide again. So I go through prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. Everything is still the same. In metaphase 2, I'm lined up in the middle. In anaphase 2, I'm pulling them apart. And then finally, in the very, very end, I'll now have 4 cells. And every single one will be different. Maybe the blue one went over here. Maybe this is the blue and red one. Right there. Maybe this one's the pure red. Maybe this one's the red with a little bit of blue. Like that. So my goal is genetic variety and haploid. How do I make sure that I'm haploid? Well, I just divide again. If in mitosis, if I divide once, if I have identical copies, then if I want half as much DNA, I just divide again. If I wanted like a fourth as much DNA, I would divide again. If I wanted an eighth as much, I just divide again. I just keep dividing over and over and over again until I get the amount that I want. So if I divide twice, then I have haploid cells. Are any of these chromosomes identical to each other anymore? Nope. What caused them to be different? What event caused them to be a little bit different? Crossing over. So crossing over gives me genetic variety. So now, if this is a dude, all of these are sperm. If this is a lady, all of these are eggs. And they're all unique. So I could now have some animal, picture like a fish. I have no idea how many eggs a fish lays, but I've seen Nemo, and that's probably accurate. So a female fish, maybe she lays like 400 eggs. And then fish are gross, so this dude just kind of swims by and like squirts some sperm out. And you're like, that's kind of disgusting. And you're like, I've walked through like a little stream before, and you're like, yeah, what was in that water? Exactly. Fish love was in that water. If upstream, it's like spawning season or whatever, then there's just females squirting out eggs everywhere. There's males squirting out sperm everywhere. And then somebody else is over here, and they're like, can we drink this? And you're like, no. And they're like, it looks clear, and they slurp it up. Disgusting. So, same thing over here. You might lay 300 or 400 eggs, and they're all just slightly different from each other. Those get fertilized by 300 or 400 different sperm that are all slightly different from each other. So you could have 400 kids, and they're all very slightly different from each other. Somebody can probably handle whatever's going to happen in the future. Okay, so far, with meiosis, the two biggest things are crossing over happens, and then in order to be haploid, we kind of divide twice. So if you were to look at this, the diagrams get kind of messy. but they basically just label them like 1 and 2. So if I was looking at meiosis pictures like this, prophase still looks the same. Metaphase still looks the same, but now I can see that they've like crossed over, but I still line them up in the middle. I still like pull them apart in anaphase. And so that's like prophase 1, metaphase 1, anaphase 1, telophase 1. And then when it's time to divide everything again, I just do it all again. And so now this time, on my way through, it's just called like prophase 2, metaphase 2, anaphase 2, telophase 2. Like that. So here's prophase 2, metaphase 2, anaphase 2, telophase 2. It's still, everything just lines up in the middle in metaphase. Everything just lines or gets pulled apart in anaphase. So my goal is genetic variety. I got that from crossing over. And then my goal is to be haploid so that I don't have freaky kids. Nobody wants freaky kids. All right, good with that so far? There's only one more thing to know about meiosis. Actually, two more things. One is kind of and then one's really interesting. One explains like how Down syndrome and other trisomies happen. So there's one other way that we get genetic variety. So ways that we get genetic variety, I'll kind of say like, so genetic variety, crossing over causes genetic variety. And then also, independent assortment. That also gives me genetic variety. Independent assortment. So crossing over gives me genetic variety, and so does independent assortment. Blue, red, blue. Independent assortment. Okay, so I've got a chromosome 1 from mom and a chromosome from dad. I've got a chromosome 2 from mom and a chromosome 2 from dad. So this is where like that third arm would have been awesome. So I've also got chromosome 2s from mom and 2s from dad. I've also got 3s and chromosome 4s and chromosome 5s and 6s. So here you go. I go through an S phase. Now I'm going to copy everybody. So the two green ones are joined together. Those are chromosome 2s from mom, let's say. These are chromosome 2s from dad. The blue ones, that's chromosome 1 from dad. Chromosome 1 from mom. Okay, so now, they would be like lining up just like this. So just like I have blues and reds side by side, blacks and greens would be side by side. My black markers and green markers would be side by side also. These guys do crossing over, just like in our picture. So these guys also do crossing over. The green ones and the black ones, my chromosome 2s, also do crossing over. Everything's the same. Same thing's happening in chromosome 3 and 4 and 5 and all that. They do crossing over. All right, so far everything is the same. Independent assortment. Independent assortment says that chromosome 1s don't influence how chromosome 2s line up. So for example, right now, they're lined up this way. So eventually, they're going to split into like 4 cells, right? So right now, what colors are going to end up in this far left cell? It'll be this guy, pure red, and then this marker, pure black, right? So I get a chromosome 1 from this one, and my chromosome 2 would be this one. Independent assortment says this could have happened instead. Now, who's going to end up in that cell? The red one and the green one. So chromosome 1s sort independently from 2s. When my 1s are all lined up, my 2s don't line up in like a certain way below it. They could have lined up this way or this way or this way or that way. It's just random. So chromosome 1s sort independently from chromosome 2s, which sort independently from 3s, which sort independently from 4s. So that also gives me genetic variety. The reason that matters is this is why there's no correlation between your height and your predicted outcomes in AP biology. Because height is likely on one chromosome. Intelligence is likely not on the same one, so they have nothing to do with each other. Otherwise, the way I would recruit is I'd be like, hey, as long as you're taller than 5'4", take AP Bio. Why? Well, people that are taller, like they obviously are more intelligent. That correlation isn't there. There's no correlation between your height and your hair color. There is no correlation between my IQ and, pick something else, like the length of my forearms or something bizarre like that, because genes generally sort independently. One gene doesn't influence another. So that's why, in general, you can't look at somebody and guess their IQ. Again, there's some weird exceptions. Sometimes I can look at somebody and tell that something's not quite right. Like, oh, that person seems like they have this genetic disease. Therefore, I know their IQ is lower. But if you just saw the stuff that's on my driver's license, like what color eyes do I have? What color hair do I have? How old am I? You can't be like, well, that guy's going to be dumb. Why? You're like, you've seen his hair color, right? That guy's, I bet he can't do math or something like that. So most genes sort independently. However, are there predictions I can make about your eye color based off of your hair color? All right, so let's say that we've got 3 hair colors. We're going to say dark, blonde, and red. So anything that's dark, so if it's like brown to black, we'll say dark. And for eye color, we're just going to say dark and light. So you have brown or you have something that's light, like green and blue. Okay, so random chance. Random chance would say that if you have red hair, you have a 50-50 shot of having dark eyes or light eyes. That would be random chance. But if you have red hair, what color eyes do you have? Yeah, they're light. They aren't dark. If you have black hair, what's the likelihood that you have light-colored eyes? Pretty slim. It's not impossible. So some genes are linked to each other. So sometimes I can predict something about you. If I know this one thing, I can predict, and it's statistically significant. Oh, you have red hair? All right, I'm going to go ahead and go out on a limb, and I'm going to guess that you have fair skin, lots of freckles, blue eyes, and pretty good chance you're Irish. And I'm right most of the time. Not all the time, but most of the time. So this is why, if I go to the doctor and they ask what race I am, of course, once I walk in there, if I'm talking to the doctor, they can tell. So I can be like, I'd rather not say, and they'll be like, okay, come on in, Tom Smith. I'm a doctor. I can figure this out. So, but the reason they ask you that is different gene pools have different genetic defects that run in their gene pools. For example, I'm white, so I have a 4% chance of carrying the cystic fibrosis gene. The darker my skin is, the less likely that is to be in my gene pool. The darker my skin is, though, even though that means I'm protected from cystic fibrosis, the darker my skin, the more likely I am to carry sickle cell anemia. Oh, you're Jewish? We've got Tay-Sachs for you. So whether it's race, religion, whatever, we've got a disease for you. So if I go into the doctor and I'm like, I've got these symptoms right here, and they're like, oh, do you have Irish heritage? And you're like, yeah. Okay, cool. Well, here's likely your issue then. And the Irish gene pool is this. Or if I'm like, hey, I've got these symptoms. And they're like, oh, well, what country is this patient from? They're like, oh, they're from this country. Oh, well, this is in that particular gene pool. So knowing my race and my sex for medical reasons helps. If I go in there and I'm like, I've got these symptoms, and it's like, sounds like breast cancer. And you're like, it's a dude. Okay, possible, but all of a sudden, breast cancer is much less likely. So that's why they will ask you on your medical forms for what race, sex, stuff like that. So anything you can give them that's useful. So you might be kind of like me. I'm a mix of several different things. I'm not just all purely from one country, but I'm mostly European, or maybe exclusively, I don't know, but mostly European. But if you're like a mixture of 2 or 3 different things and you know that, you can put it on there because certain things run in certain gene pools. So the goal of meiosis is genetic variety and haploid. How do I get variety? I trade DNA, and then genes sort independently. So as a general rule, Genes sort independently. Some genes are linked, like my hair color and my eye color. But most genes are not. I can't go and recruit the basketball team based off of hair color. Height, that might be an advantage. But otherwise, all right, questions on that? The main way in any college class other than like developmental biology that you see meiosis is they're always asking you to compare it to mitosis. They're always looking at what's different between the two. So it's mainly haploid, crossing over, independent assortment. They do have some extra vocab that they can throw at you if they want to be mean. When these guys line up side by side, that's not the biology term. They call it a tetrad. Over here, they don't line up side by side, so that's not a tetrad. What number does tetra mean? It means 4. 1, 2, 3, 4. These guys don't line up side by side. They line up head over heels. These guys line up side by side. The science way of saying that is the chromosomes line up side by side and they form a tetrad. 1, 2, 3, 4. Over here, we don't form tetrads because they line up head over heels. Obviously, no textbook is going to call it like head over heels. They just call it not a tetrad. All right, so whenever we start looking at this, you're like, okay, well, how does something like Down syndrome happen? Remember, when we looked at Down syndrome, how is it that I get like 3 chromosome 21s? Or how do I get like trisomy 10 or something like that? So if I have Down syndrome, I've got an extra chromosome 21. Instead of 1, 2, I've got like 1, 2, 3. So how does that happen? Let's say this is Dad right here. And right here, this is supposed to split. These two sister chromatids were supposed to split and go into 2 different cells. What if the chromosomes split apart, but they didn't get split into 2 different cells? What if like they both ended up over here? Like this. What if both of them ended up in the same cell? And let's say that these are chromosome 21s. How many chromosome 21s does this future sperm have now? 2. What about this one? None. Normal, normal. So let's say that that sperm goes and fertilizes a normal egg from a female. And she's got 1 chromosome 21. When that sperm fertilizes that egg, how many chromosome 21s do you have? 3. And so that's how trisomies happen. What if this sperm fertilized that egg? That's how a monosomy happens. Now that's how you have only like 1 chromosome. For example, if I have trisomy 21, that's Down syndrome. But if I only have 1 X chromosome, I can survive. I'm a female and I have what's called Turner syndrome. So how does Turner syndrome happen? Well, if that's the X chromosome and then this one got a nothing, that would be Turner syndrome. So they have a name for this. And at first, the name sounds stupid, but the name actually makes sense. If these guys like don't separate properly, they call it non-disjunction. 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 So I'm trying to translate non-disjunction. What does that mean? Doesn't that basically just mean junction? There's junction. Doesn't disjoin mean like take apart? So wouldn't non-disjunction, wouldn't that be like, so disjunction means don't join. Doesn't this mean like don't join? So doesn't non-disjunction just mean stay joined? So the reason they say that though is right here, weren't these 2 chromosomes, this guy and this guy, weren't they supposed to like disjoin into 2? But did they? No, they nonned. They were supposed to disjoin, but they didn't. So they both went over here, non-disjunction. So the name actually makes sense. They stayed together. They were supposed to disjoin, and they didn't. So that's how trisomies and monosomies and quads and whatever else happens. How do you end up with more than just one chromosome 2 from dad and one chromosome 2 from mom? Non-disjunction. Anytime you don't have pairs up here, it's always because of non-disjunction. All right. Feeling all right on meiosis? Cool. Mendelian genetics, Mendel, or Mendelian genetics. So I'll start off with a picture of this guy. Students always find this guy interesting. So what's interesting about that guy? The world's greatest piano player. Just kidding. They're like, what chord are you playing right there? And you're like, you can't play it. You have one finger here, another finger here, another finger here, your other finger here, this finger here, another finger there, another finger there. That's how you make that chord. I play a 7-string guitar, and I can do a chord with no open chords. I have no idea if that's real or fake. However, I do know that this one is real. So that's a newborn baby. You can see the gloved hands kind of holding his hand. And how many fingers does that baby have? 6. So this is called polydactyl. You'll never have to know the name of it, but some of you guys are interested in like a medical career. Poly means many, and dactyl is fingers. Like terry means fern-like or feather-like. So a pterodactyl, it's got like sort of like fern-like wings. It's got giant fingers. Like a pterodactyl has 5 digits just like we do. It's got a thumb and then 4 fingers, and they just all extend out really, really far, a pterodactyl. So a polydactyl just means I have many fingers. Now, we always have many fingers. We should have like 5, and on our toes, 5 and 5. But if you have more than 5, then you're called polydactyl. So this child, as is common, there's multiple reasons you can be polydactyl, but the most common one means you're polydactyl on all limbs. So for him, he'd be 6, 6, 6, and 6. So hands and toes. All right, so let's say that this is my son or daughter. Does that 6th finger look useful? Does it look independent? Like I could play the piano better one day, or play another chord on the guitar, or something like that, or get an extra grip on like a ball? No. Basically, this finger is just it's just sort of joined to this one. So if this one moves, that one moves. It doesn't move independently. It's just like if I just sort of like taped this pen cap to my hand. They're like, hey, look, another finger. And you're like, that doesn't do anything. Like if I wave, I've just got this extra pen cap. It's just like taping it to my hand. Same thing with the toes. How comfortable are shoes going to be? They're not. So you just cut it off. So the kid's born, and eventually they'll have a teeny tiny little scar on the side of their foot and a little scar on their pinky. So polydactyl. I've had 3 students that have been polydactyl in my career. My very first year teaching AP biology, I even remember the girl's name, Samantha. It was 2001, probably November, because that's when I'd be doing Mendelian genetics. And she's like, hey, Mr. Cotton, I was born polydactyl. And I'm like, don't be rude. Maintain eye contact. Don't look at her hands. Don't look at her hands. Keep looking at her face. Keep looking at her face. And she goes, you can look if you want. I'm like, okay. And so her extra digit had been on her thumb. And so she just had this little bitty scar right there. Otherwise, you would never notice. You're like, how have you not noticed that she had like 6 fingers or something until then? Like at some point, you're like handing out papers and somebody's like, you know, like that. And you're like, oh, wow, there's a lot. So I had to go back and see how that came out in the video. Let me grab that paper. You know, thank you. Like, whoa, that seemed like many. I'm I'm assuming most people that are born polydactyl have a good sense of humor about it. Otherwise, that was a really mean thing to do. So anyway, she just had this little bitty scar, and that's it. And if you just looked at her hands, you couldn't tell. But then after that, she's like, see how like this, how just the angle of stuff is wrong. And it was a little bit more like for us, we have opposable thumbs. These are kind of going in this plane, kind of like the x-axis, and these are like in the y-axis. If you hear about animals that don't have opposable thumbs, that means their thumb still only goes like this way. It'd be just like having another. Opposable means one goes horizontal and the other goes vertical. That means we can grip stuff. Otherwise, like gripping something like that, I can grip stuff. But if I can wrap my thumb around it, I can like hold more weight. If I have to grip something like this, I can hold a certain amount of weight, like if I'm doing deadlifts or something. But if I can like wrap my thumb under it, I can hold more. So it allows us to grab stuff. So what I noticed, though, is if she held a pencil, like if they're taking a quiz, she holds a pencil, hers wasn't quite as opposable looking. Like her thumb knuckle, instead of being right there, would be about right here. So it looked more like a claw, like that. When she was holding it, her thumb knuckle was down like that. And I know this because every time they took a quiz, I try to nonchalantly stare at the way that she was writing. I'd like walk around, you know how teachers do sometimes. I'd walk around, and so I'd just be like, then I'd get to like Samantha's desk, and I'd just be like staring at her, writing with her quiz, or just fascinated by like the angle of her hand. Hopefully she never noticed. So anyway, polydactyl. Turns out polydactyl is dominant to having 5 fingers on each hand. So what does dominant mean then? Does dominant mean most common? No, I teach almost 200 students a year, 26 years. I've had 3 students that have told me they were polydactyl. Every single one of them, by the way, had the extra digits cut off. They were just like really, really small extra fingers or toes or something like that. So otherwise, I would have never known. And even when they show me, they'll be like, yeah, see right there, and there's just this teeny tiny little scar. Some people, the way their thumb joins is more different than others. So I kind of make the analogy of this. Let's say I go to the cafeteria. I'm never in your all's cafeteria. But let's say that I've got a chair and, you know, pretend that we're at a crowded place. So my wife and I are at a crowded place. And so she's my wife, so we could like share a chair. But if you're like sharing a chair with somebody, then you could be like, all right, you put your butt there, I put mine here, and I kind of lean this way. You kind of lean this way. You know what I mean? You could do that. So that's kind of what her thumb was like. Instead of it going straight up and down, I kind of have like a thumb going that way and a thumb going that way. And if you cut off one, then now like you've got a thumb that kind of, the angle's weird. Just like, how do you sit in a chair? Cool. How do you sit in a chair if you're sharing it with 2 people? Cool. Now cut out the other person, and this is how you sit in a chair. You're like, hey, like that. So the angle just looks off. So polydactyl is dominant. This is also due to mutations in the sonic hedgehog genes. That one's more complete. That's not just like a little bit of a digit. That thumb kind of goes almost all the way down. So that really could be an almost like useful second thumb, maybe. This is legit. This is from like a radiological society page or whatever. So that's probably the best foot I've ever seen as far as being polydactyl. You can tell where the skin is. There's like slight shadows. So like if I was wearing flip-flops and you have that thing that goes like between your big toe and your next toe, that would stop right there. So this goes all the way back. That's the arch of their foot. So it's not like, hey, you have this little like extra thing hanging off. Like if I was wearing flip-flops, if somebody was wearing flip-flops right here in the front row, and I walked by, I'd just be like, hey, what's up? And you're like, oh, cool. And you'd be like, what the, like, at first everything would look normal, and then you're just like, that was too many. But like they've got a complete extra toe. This splits way back here, like up there. So they have like an extra pinky toe. That same person, this is what their hands look like. That is a 6th finger It's a separate finger all the way up to the wrist. That's the wrist bones. These are the ones that are like the palm of your hand. That's the wrist bone. So they have a complete extra finger all the way back to there. And because of the sonic hedgehog genes, anytime you build a finger, you build a tendon and a muscle attachment. So that person does have a sixth finger. They really are a 20% better, exactly a 20% better guitar player. Proportions joke. Sometimes it can be kind of a mess. My point is this. What does dominant mean then? If 6 fingers on each hand is dominant to 5, what does dominant mean if dominant's not most common? There's this type of dwarfism that's dominant to regular height. Dominant does not mean most common. So what's the word we use for most common? Or what does dominant mean? Either one. Yeah, Addie.
That if your parents pass the genes, it means you have it.
Yeah, dominant means that it will show up full strength every time. So dominant just means this. If I inherit a gene that codes for black hair or green hair, pretend we could have green hair or fur or something. If I inherit a gene for black fur and green fur, and black shows up, black is dominant. But green might be the most common out in the gene pool of life. So for example, capital F, that's 6 fingers on each hand. Whereas lowercase f, that's 5 fingers on each hand. However, out in nature, there's like one of these for every like 5,000 of these. So which one's more common? That. So it's almost like this. Pretend that this is the gene pool of life. This is not some sort of commentary on humanity that I'm using a trash can. So this is a gene pool for life. And pretend I throw in 1 black marker, and I throw in 5,000 green ones. And this represents the gene pool, your parents. You don't pick your parents. So you're going to get a chromosome 1 from dad and a chromosome 1 from mom, and that's going to determine how many fingers you have on each hand, let's say. And I have no idea if this is on chromosome 1 or not. So I'm going to reach in there, and I'm going to pull out 2 pens. There are 5,000 green ones and 1 black one. When you reach in there and pull out 2, what are you going to end up pulling out? What colors? 2 green ones. And so you'll have 5 fingers. Throw them back in. I go to the next student. Take them out. What are you going to pull out? 2 green ones. Or picture they're like Scrabbles, kind of like Scrabble blocks. There's 1 capital F and 5,000 little F's. And you pull out 2. What did I get? Little f, 5 and 5. Throw it back in. What did you get? Little f, 5 and 5, and so forth. One out of every 5,000 people, though, they're going to pull out a capital F and a little f. Capital F is dominant, so how many fingers will they have? 6. So dominant just means shows up full strength every time. This is the first time I've ever said write down a definition exactly as I say it. So I'm going to have you somewhere, and I've got it buried in your notes somewhere, but dominant, write down, shows up full strength every time. If you define dominant that way, that helps you with a bunch of other stuff in the future. That's why we've been together for like almost a semester, and I've never said define this exactly the way that I write it, ever, until now. So dominant means shows up full strength every time. So if I have a dominant gene or whatever, that means it shows up full strength every time. So if it codes for red, it's full strength red. It doesn't show up as like medium red. It's full strength red. Shows up full strength every time. That's what dominant means. Does anybody know what the word is for the most common thing in nature? 5 fingers on each hand is not dominant, but what do we call it? Wild type. Nice. Wild type is the most common type in the wild. So 5 fingers on each hand is wild type. But what's dominant? 6 fingers on each hand, 6 toes on each foot. So wild type is the most common. Sometimes it's hard to tell. Sometimes there is no dominant one. Like hair color. Yeah, worldwide, darker hair is probably more common, but it's not like 99%. There's like quite a variety. Yeah. No, it doesn't necessarily mean that it has to be a mutation. The way it is, but the reason that we know it's a mutation is because like my anatomy is not set up for a sixth finger. Like for example, my wrist right here, I know you haven't looked at like very many wrist x-rays in your life, but can you tell how like that bone seems to fit with the wrist? That bone seems to join okay. Can you see how here, how it looks a little crowded and like that guy's kind of fallen off? Almost like that chair is supposed to hold two people, or that bone is supposed to hold two fingers, but it's like there's a third sitting on there. That's how we would know that it's a mutation. So it's possible that whatever the wild type is could be a mutation. So for eye color, anything other than dark eyes is a mutation. But blue eyes and green eyes are still pretty common. Maybe not as common as brown, but still pretty common. Like in this class, I bet half of you guys have brown eyes and half of you guys have something else. So it is possible that the more common type could actually be a mutation. But you're right. Typically, wild type is the one that's like not a mutation. Sort of weird. So there's a type of dwarfism, like I said, that is dominant and regular height is recessive. Achondrioplastic dwarfism. Kind of strange. Back to those sonic hedgehog genes. Normal wild type mouse. Mutate the sonic hedgehog gene. There you go. In humans, sonic hedgehog gene mutations look like 6 fingers or 5.5 fingers or some sort of like digit mutation. In mice, it looks the same way. They also have like limb deformities if they have sonic hedgehog mutations. So weird. So dominant means shows up full strength every time. All right, let's look at one more thing and then we'll be done for the day. Let me take rolls super fast. I don't know why I always say super fast. We've got, nope, I'm missing Lucas. That's it. All right, so I've got this picture up here, and this is Mendel. You guys remember Mendel, Gregor Mendel? He was a monk, and so when you're a monk, like I've never been a monk, but I think you have a lot of free time. Like you don't really have very many possessions. You know, you get up, you pray, and then I think after that, like your day's pretty much just like wide open, more or less. And so he started doing experiments with plants to figure out stuff about genetics. And so here's what he did. This was in like the late 1800s. And so this is around kind of the time that Darwin was around. And whenever he did this, his stuff was kind of so ahead of the times that nobody gave a crap. Like he published his study and nobody cared. It was a complete flop. Then like years later, all of a sudden somebody's like, oh, this guy was like so far ahead of his times we didn't realize his genius. So here's what he did. He'd take a bunch of flowers and breed them with each other. So with flowers, you have male parts and female parts. Like there's a flower, that part in the middle right there. Those are the female parts. These are the male parts. That purple stuff is pollen. So pollen will land on the sticky stigma. Sperm will sort of hatch, so to speak, from the pollen. It'll swim all the way down there. And down there's the ovary. It'll fertilize the eggs. The eggs become seeds. And then the ovary becomes fruit. And then we eat it. We eat the ovaries of plants because they taste good. There's pollen. We want to get bees and flies involved. We make something sweet, like nectar. They have little nectar factories, literally called nectaries. And then we rub pollen all over this bee, which is sperm, and so that's gross. And then this bee will go fly around, and he'll end up spreading the pollen from one flower to another. So he's helping them. It's like this weird like plant-bee love triangle where he helps them pass on their offspring. Those are the eggs. So pollen lands on this sticky stigma. It swims down here. That's the ovary. And it's full of eggs. Just like in a human, you have two ovaries and they're full of eggs. And sperm swims down a canal. So sperm will swim down there. If one sperm fertilizes one egg, then this future fruit will have one seed. If 50 sperm fertilize 50 eggs, then this future fruit will have 50 seeds. So one apple that I'm eating has like 10 seeds. Another has 12. Why? That's how many like eggs got fertilized. That's so bizarre. Okay, so what Mendel did, which I think is ironic because he's a monk, is he would like force which flowers were going to breed with which flowers. So he'd like break off the male reproductive parts and like rub them on the female reproductive parts, all after he like prayed to God, which is kind of weird to me. So he does that, and this is the data that he gets. So here's what people had done before Mendel. Before Mendel, people had like bred stuff. They were trying to figure out genetics. All we really knew was that like, hey, kids tend to look a little bit like mom and a little bit like dad. So some people had done experiments with plants before. And here's what they would do. They'd be like, all right, I'm going to take a red flower plant and I'm going to breed it with a white flower plant. The reason I did red and white is because I know that if I mix red and white, I know the intermediate color would be like pink. And so what were they expecting when you breed a red flower plant with a white flower plant? You're expecting pink. What did they get? 100% red. So these are the parents. These are their kids. We'll talk about it later, but they call it the F1 generation. They expected pink, and they got 100% red. This is just as red, just as dark red as that. And they're like, huh, that's weird. And they moved on. Some scientists were like, all right, what if I breed this red one with its brothers and sisters? They're flowers. They don't know any better. But what if I take some of these kids that are red and I breed them with each other? So I'm breeding red ones with red ones. So what would I expect if it's like 150 years ago? If I breed red with red, I would expect red. What did I get? Red and white. So if I breed red with another one of these red ones, not this red one, but one of these red ones, then I get red and white. And nobody could figure it out. And then Mendel kept numerical data on this. And here's what he found. So over here, when he has the parents, the F1 generation, that's their kids, these were 100% red. So these guys right here were 100% red. These over here, you got a mixture of red and white. So he did this with like flower color, seed shape, pod color, stem height, all these things. What do you notice about those numbers? The word dominant and recessive didn't exist yet. He invented those words. The word dominant in like English existed, but like the science word dominant didn't exist. Is there anything that you notice when you look at those numbers? This is how he figured out dominant. Mendel is the guy that basically figured out dominant and recessive. So kind of like right here, I got a mixture of red and white. So pretend instead of purple and white, that said red and white. Do you notice anything when I compare this side to this side? Dominant seems to show up more. I agree. What else? More detail. Same idea, but more detail. How much more? Three times more? 2,000 to 6,000, that's a 3-to-1 ratio. 705 to 224, pretty close to a 3-to-1 ratio. That to that, it's about 3 times bigger. It's a 3-to-1 ratio. 3-to-1 ratio, 3-to-1 ratio. So he's like, huh, seems like it's always a 3-to-1 ratio. So when he starts looking at red versus white, it was like 3 to 1. So he's like, oh, I think I've got it. There must be this thing called dominant. Because here's what happens. When red and white breed, it looks like white disappears. But did white get destroyed? Nope, because it comes back. And it's full strength white. It's just as white as that one. So white is still there, it's just hidden, it's masked somehow. So what if, the reason that we kind of look like mom and we kind of look like dad is, what if we get 2? We now call them genes. What if we get 2 genes for a color? So maybe that guy's big R, and he's dominant. And this guy is little r, and he's recessive. This other dude named Punnett came along and said, I've got this square. And so they're like, all right, all of their kids would be big R, little r. And since red is dominant, they would all be red. Then if I take their kids and I start, you know, brother and sister together, these are flowers again. They don't know. As far as we know. Turns out flowers can contract like muscles, and they can send electrical messages just like our nerves work. So maybe they do know. Maybe they're like, Mendel, what are you doing? So over here, red, 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 white, 3-to-1 ratio. So he basically backward engineered. Based off of this, he came up with the idea of, hey, we get 2 versions of genes. One's dominant and one's recessive. And that's how it works. So this is Mendelian genetics. Now, does everything in life work that way? Like, aren't there pink flowers? Aren't there some things where if you take a red and a white, it shows up pink? Yeah, so we're going to learn 8 ways to break Mendel's rules. Human skin color, for example. There's a variety. So if I'm a dermatologist, I think they classify it on a 6 scale. So your skin darkness or lightness is a 1, 2, 3, 4, 5, or 6. So if I have somebody that's like a 6 and somebody that's a 1 and they have kids, if dominant recessive was the thing, then their kids would either be like a 6 or a 1. But aren't they typically somewhere kind of intermediate? So if I have a dark-skinned parent and a light-skinned parent, then aren't I kind of like in the middle? So Mendel's rules don't work with everything. Mendel's rules work with some things. These are Mendel's rules, and then we'll look at how to break Mendel's rules. So that's it for today. Oh, one last thing. What was our definition of dominant again? It shows up full strength every time. Yep. All right. That's it. See you later.